and taking the time to talk to us about this most important topic. And as you and I have traded emails over the last couple of months, I think I've shared with you that this, I think, is one of the most misunderstood parts of beekeeping that there is. I think the range of understanding of it is everything from it's kind of a sick hive to, it has great benefits to a hive. And so we're anxious tonight to hear what you have to tell us about that. And just in the spirit of transparency, we do record these, and we do then post the recording on our website for members to come and view later. And they've become a valuable library to our beekeeping members. So we just want to make sure you know that.
That's great.
Yeah. Also, Kurt sent out a chat to everybody that that the questions and answers should be posted in the Q&A section of the Zoom window. So don't put them in the chat because they're not likely to get answered. So if you would put them in the Q&A. So Dr. Spivak, I told you we love to hear a little bit about your bio from the biomaker themselves. And so if you would just take a minute and share with our group what brought you to this part of your life, and a little bit about your background, your educational experience, and your research, and all of that.
All right, thank you. Thank you, Scott. And hello, everybody. Good evening. I know it's an odd time for you to have this, but I appreciate you accommodating. Let's see. While we were gathering, I was telling probably a story I probably shouldn't have told about, but I got into bees when I was 18 years old, and through reading a book. I'm not sure I recommend the book. It's called Bees Ways, but it caught me. And I went to work for a commercial beekeeper, and I got a bachelor's in biology at Humboldt State, what was then called Humboldt State. It has a Cal Poly, I think it's called now. And then I got lost in South America for a couple of years, traveling and searching for beekeepers. And down there, I was in Venezuela for a little bit, assisting Dr. Orly Taylor from University of Kansas as a beekeeper for some projects he was working on, and then he asked me to be a graduate student. So I got my Ph.D. from University of Kansas in 1989, did a postdoc at the Tucson Bee Lab and the University of Arizona, and then a job posting came up just at the end of my postdoc, and I was getting a little desperate to find a job, and they posted a job for non-tenure track, 3-year temporary position at University of Minnesota. And I thought, I had promised myself I would not live in a cold climate or a big city, but I said, oh, I can do anything for 3 years. And it's 30 some years later, and here I am. I just retired January 1st. That's exciting for me. And we've endowed my position, and applicants are in, and I'm excited to see who will replace me. So that's the story, I guess, in a nutshell. And I'll share my screen now. Let's see. I need to move some things around here. Okay, so I can see. So the title of this talk is Why Care About Propolis? And I'm going to pronounce it Propolis, but I think Propolis is fine too. Tomato, I don't think it really matters. And I hope my goal is to convince you that it's actually really good stuff. It's important for bees, for their health. And even though it's extremely sticky and it's on all of my clothing, I hope that you learn to love it. Let me see. Okay, so what is propolis? It's actually plant resin, and probably through this talk, I'll use the resin and propolis interchangeably. They're the same thing. So some plants, not all plants, secrete resin, very sticky resins, as a plant defense. It's a secondary plant compound. It helps protect the plant from herbivory, from infection, from UV radiation. These resins are highly, highly antimicrobial. That means anti-bacterial, antifungal, antiviral even. And honeybees are able to, well, other bees too, but honeybees tonight are able to locate these plants that produce these resins, secrete them, and they go and scrape them off the plant. The left-hand photo is of a bee in Brazil collecting green propolis from Baccharis. It's a shrub. And they pack it on their hind legs, like the second photo with the yellow propolis on her hind legs. And then it's difficult for the bees to get it off of their legs on their own. So a nestmate often helps tug the resin load off the hind leg of the forager. With her mouth part, she pulls it off. And then together they cement the propolis into the nest cavity. So from the plant, it's resin. And once it's in the bee colony, we call it propolis. The bees, as far as we know, are really not changing the chemical composition of the resin. And that's why I use the terms interchangeably. They can add beeswax to it. They can add some enzyme secretions. But when you look at the chemistry of the resin from the hind legs of the bee and the propolis inside the nest, they're basically identical. So they're not modifying it like they might modify honey somewhat. So here, I hope this works. It's a video that Christine Kurtz, a beekeeper, sent me. I think she's from California. And it's going to go slow-mo. And about 30 seconds in, you're going to see this bee. She's actually stealing propolis from the front of a different colony. I don't know if you can see my pointer, but on her hind leg, there's a little yellow blotch of resin. And she's going to pull some of the propolis, resin propolis. She's going to pull some with her mouth parts from the entrance of this hive. And it's going to happen right about now, coming up. And she's going to transfer it from her mouth part right now to her front leg. That's what's happening. And then she's going to transfer it to her middle leg. And then that's happened. And then the middle leg scrapes it off and tacks it to the outside of her hind leg. That's also how they collect pollen. But I really love that video. All right, so once they come back with these resins, if they live in a tree cavity, a natural tree cavity, like shown on the right, they'll line the whole interior of the tree cavity with a layer of this propolis. So this is a figure from Tom Seeley back in '76, a book he wrote. And the photo on the right is a bee tree that someone gave us that we opened up to see I wanted to see the propolis envelope. The bees unfortunately had died. And so we took out the rest of the combs, and all of the black, dark brown stained area in the interior of the tree is the propolis lining. Oh, wow. Sorry, I hope it's not on automatic, but that was advancing without me. So everywhere below that red dotted line is the propolis envelope, and you can see above the line, you can see some mold and other stuff growing. And it kind of tells you everything right there, just looking at it, that it's really there. It's sterilizing the interior of the nest cavity. Okay, this talk's going to have a mind of its own. Sorry. Humans have had a long interest in propolis. It's actually referenced in the Bible, not as the word propolis. I forget what the word is called, but they have used it medicinally. Humans have used it medicinally for as long as we have recorded history, I think. It's because it's so antimicrobial. It's active against mostly gram-positive bacteria, fungi, and tumor cells and viruses in humans. It can enhance the activity of ointments, antiseptic washes, and some research that was done at the University of Minnesota, which is one of the reasons I was pulled into the world of propolis to study it, is that they found, at least in the laboratory in cell cultures, that propolis was active against HIV. So it was preventing the entry of the virus into the cell. So I thought, wow, if it's so, that's really amazing. If it's so good for human health, it's got to be good for bee health. There had been some studies in the laboratory of the activity of propolis against varroa on its own in a petri dish, against chalk brood, the Ascosphaera apis, the fungus that causes chalk brood disease, and against the bacteria that causes American foul brood. But all of these were done in a laboratory, and the questions we were asking was, does it really function in an actual bee colony? What's it really doing? So just to give you some context, the bottom two photos are that propolis envelope within the tree cavity, and the bottom right photo is that same tree I showed you, but you're looking out the front door as if you were a bee looking out the front door. And that photo shows pretty clearly, especially around the entrance, how thick that layer of propolis is. The orange, yellow, brown, reddish layer is part of the propolis envelope. And in contrast, in our Langstroth boxes that are smoothed pine, the bees are not depositing a layer of propolis. They'll put it everywhere where it gets in your way, right? In the cracks and crevices and the frame rests between the boxes. And for that reason, beekeepers don't really like propolis. They like to get rid of it. And actually, there's been selection against colonies that produce propolis. So queen producers are actively selecting, have actually actively selected for colonies that don't bring in as much propolis, just so that it makes beekeeping easier. And I think that's a big mistake. I think we've made a big mistake. So over the years, these people that are listed at the bottom, who have now gone on to get real jobs and excellent careers, as graduate students in the lab, they ask these general questions. What are the plant sources of resins in our area? What's the benefit to a honeybee colony of having a propolis envelope? Does it maybe help reduce disease load? How does it affect the microbiome? If it's so antimicrobial, would it affect the beneficial or microbiome bacteria in the gut and on the exterior of the bee? And then, do colonies sell for social medicate? In other words, when they're sick, do they go collect more resin as medicine? So I'll briefly touch on all of these quickly, jumping right to the point with no methods. Well, I'll tell you briefly. In our area in Minnesota, bees are collecting almost all of their propolis from eastern cottonwood, Populus deltoides. If you go and during the summer and you grab a branch of a cottonwood tree and you smell it, especially the new leaf buds, you'll say, wow, that smells just like my bee colony. And they're scraping it right off the leaf buds all season long. They also can collect from balsam poplar. So anything in the genus Populus is a great propolis source for them. We know they collect different sources of resin in different parts of the world, but in our area, it's from Populus. And this was work from Mike Wilson. He collected resin from the hind legs of individual bees and resin from the plant. And he, with liquid chromatography followed by mass spectrometry, he found he could match the profiles, the chemical profiles, metabolic profiles of the resin. And that's how we could determine what the plant source was. In experiments, we had to make a propolis envelope experimentally. And one way we did it, shown in the boxes on the left, those little 5-frame nuke boxes, is we made a tincture of propolis. So we collected propolis from different areas, and we dissolved it in ethanol, 70% lab grade ethanol and strained it, and we could get a percent solution that we knew, and we could apply a known quantity. We could paint it right inside the boxes and then put the bees in these boxes. And then once we had them established in there, in this particular experiment, we challenged the bees with chalk brood. In other words, we ground up chalk brood mummies and mixed them into a pollen patty and fed them to the bees. And if you do that, they get chalk brood. I don't recommend you do this. This is for an experiment. And the graph on the right shows if the bees had a propolis envelope, with the bar with a red line around it, the propolis envelope around it. And this is a very small experiment. We had only 6 colonies there. But if they had a propolis envelope, the amount of chalk brood mummies we counted in that colony was significantly lower than colonies right next to them that did not have a propolis envelope. So it didn't cure the chalk brood, but it really reduced the amount of chalk brood that we were seeing. We did the same with American foul brood. Another thing I do not recommend you do. In this case, we made a sucrose, a sugar solution. Then we put bacterial spores, the bacteria Penibacillus larva that causes American foul brood. We mixed it into the sugar syrup and sprayed it right on the young larvae, and then put it in colonies with and without a propolis envelope. So in this case, the maroon colored bars are what happened. Those are the colonies that had a propolis envelope, and the gray bars are the colonies that did not have a propolis envelope. And we measured the amount of American foul brood they had over time by giving them a severity score. So 1 is, we could see 5 cells per each side of the frame, for example. And you can see that, yes, they got American foul brood, all of them, but if they had a propolis envelope, they had significantly less. The disease load was reduced. So it was helping them out. It's not a treatment. It just helps the bees fight the infection. In many experiments, we also looked at the immune response of the bees, and this will be clear why we did that coming up. But for the propolis experiment in this particular case, we tacked propolis traps inside the colony. And the photo you're looking at, the propolis envelope top left, is we had removed the traps just for the photo. During the experiments, we left them in there. The bees, propolis traps are these plastic grids with 1/4 inch gaps in them. They're about a little over 1/4 inch deep. And at that width, bees stuff the cracks with propolis. It really interferes with the bee space, so don't do this. Don't do this at home either. But we put bees in colonies with this kind of propolis or controls, just regular Langstroth boxes, and marked bees and removed them from colonies when they were 7 days old. And we did some real-time PCR and looked at all of these crazy names here, Hymenoptacin, Abacin, Defensin, which are all either antimicrobial peptides or part of the bees' immune systems. And if you look at the maroon bars, in every case, they are lower than the gray bars. Whether the graphs go up or down, they're kind of crazy data to look at. But this is showing that the bees' immune response was always lower if they had a propolis envelope. And that seems like it's not a good thing, except I'll explain it in a minute. If they had a propolis envelope, the other thing is that their immune systems were stable over time. The variation in the gene expression was less if they had a propolis envelope compared to bees without a propolis envelope. So their immune gene expression was lower and more stable throughout the season. And we think this, I'm pretty sure this is because the propolis envelope is killing off opportunistic and pathogenic microbes within the nest cavity. And when that happens, the bees don't have to activate their own immune systems as much. The propolis is doing the job for them. So the propolis is, in some sense, helping to sterilize or get rid of some of the other microbes in the nest. and the bees don't have to activate their immune systems. It's costly for bees and all organisms to activate an immune response. So it's a good thing to have a quieter and more stable immune response. And that's another benefit of the propolis envelope. We did a couple of studies looking at the microbiome. Again, we looked at bacteria only, not the fungi that are in the microbiome. But with propolis being so antimicrobial, our question was, does it affect these beneficial bacteria that are part of the microbiome? And in this particular study I'm showing you, done by Holly, we looked at, and in collaboration with Kirk Anderson from the Tucson USDA lab, we looked at the mouthpart microbiome. So bees, of course, the microbiome is in their guts, but it's also all over their bodies, and in particular, coating the mouth parts. And if you think about it, bees get their mouths into everything. They feed larvae with their proboscis, their tongue. They're collecting nectar from flowers with their mouth parts. They're sharing food mouth to mouth with each other. They're touching the queen, licking the queen. So we were curious what the microbiome looked like on the mouth parts only if they were in a colony with a propolis envelope or not. So Kirk did some gene sequencing of a particular section of RNA. And this graph on the left, the first bar shows the amount and kinds of bacteria that we found on the bees' mouth parts if they had a propolis envelope, or the second bar if they did not. And the green bars are the core microbiome species that you would find on a worker's mouth parts: Bombella apis, Fructobacillus, Lactobacillus. So you can see there's more green than blue, and the blue are the pathogenic or opportunistic bacteria. And you'll probably recognize some of these names: Pseudomonas, Streptococcus, Serratia, Microbacterium. These are either bacteria that are just present in the environment, and opportunistic, or disease-causing, pathogenic. And the bees in the colonies without the propolis envelopes had significantly more of these pathogenic or opportunistic mouth bacteria on their mouth parts. So the propolis, again, is helping the bees reduce that disease load or that bacterial load. And moving to the question, if bees are sick, do they go collect more resin from plants? And so, in other words, are they self-medicating? And more correctly, it would be social medicating, because the bee that's foraging probably is not the sick one. In fact, the diseases we gave the bees are these brood diseases, chalk brood or American foul brood. So the bee that's foraging for resin isn't collecting it for herself, she's collecting it for the colony, and that would be social medication. So to do this, we set up a bunch of little 5-frame nuke colonies with bees in them. And before we did anything, we would sit in front of them twice a day for 20 minutes each time. We would plug the entrance and just sit and count the number of bees returning with either resin on their hind legs or pollen on their hind legs. And the bottom photo shows resin on the left on that bee, or pollen on the right. This was work by Renata Borba and Mike Simon Finstrom. So we would count the number of resin and pollen foragers. The pollen foragers was just a control for the amount of foraging. And then after 2 weeks, and we got a good count of average number of resin foragers per colony, we challenged the colonies with a pathogen. So either chalk brood by feeding them chalk brood in pollen patties, or spraying American foul brood bacterial spores in sugar solution on the larvae. And then once they were sick, we would sit and count resin foragers again for 2 weeks. It was pretty fun. It's fun if you have good eyes, actually. And what we found, and we repeated this experiment several times, this is just 1 year in 2014, When we challenged the bees with chalk brood, which is Ascosphaera apis, this taller gray bar, the bees did send out more foragers for resin. The number of foragers after minus before the challenge increased significantly from the controls. But if we challenged them with a bacterial pathogen that causes American foul brood, the middle bar, they increased foraging for resin a little bit, but not different than the controls. And we repeated this 3 times, and it was always the same. And why bees would increase resin foraging after a fungal infection, but not a bacterial infection, is way beyond me. I have no idea. And I hope somebody follows that up someday. So summing that up, breezing through many studies, propolis is really good for bees. The antimicrobial properties of propolis provide, I will call it everyday benefits to the bees' immune systems. It promotes and stabilizes their core microbiome species and their immune systems, and it helps them reduce their disease load. So how can we all encourage our colonies to construct this propolis envelope within the colony, and maybe not have the bees put it where it gets in our way between the, under the frame rests, etc. Okay, so backing out a little bit, as you probably know in your own bee colonies, some colonies really collect a lot of propolis. It's a heritable trait. In other words, you can breed for it. It's a genetic characteristic. Some colonies, it's a genetic trait. Some colonies collect a lot of propolis and some very, very little. Often they'll stuff the cracks in your equipment, sometimes put it on the tops of the frames. Probably you've all seen it in the entrance, especially in late summer or early fall. Some colonies really like to reduce the entrance side by putting a lot of propolis in the entrance and just leaving a small little hole. Sometimes on a really warm day, you can see it dripping out the front of the hive, like on the right-hand photo. In Brazil, Africanized bees, that subspecies of bees, really collect a lot of propolis. And what they do to collect it down there is they modify their boxes so that they can remove these 1 or 1.5 inch width slats from the side of the box. And they remove one at a time. And while it's open, the bees fill it with propolis. You can see after they've removed all the slats, this is what the side looks like, side of the colony. And then they can just take a knife and cut out huge sections of propolis. And I had a student visiting from Brazil, and he said, well, we were trying to figure out how to collect propolis. And he said, well, why don't you just leave a big crack in the side of the box? And I said, I don't think that's going to work for our European bees. So we tried it anyway. He insisted. So he propped open the box, and the colony got robbed out, and they didn't put any propolis there at all. It was just, our bees just don't fill up the space like that. The Africanized bees are really impressive that way. So to make this propolis envelope, we tried all kinds of materials. We tried various types of mesh. This is a very loose mesh, like the stuff that comes in an onion bag, but finer mesh. And we tack that in. And yes, the bees stuff it with propolis, but it shreds, and it ended up in the honey supers, and there was green plastic shreds all over the colony. And that was not a great thing. We tried a harder, more durable mesh. And yes, the bees put propolis in it. And that's not a bad idea. You can't really harvest the propolis from there. But mostly we wanted something we could just leave in the colony and that the bees would collect on their own. And we wanted more natural solutions without having to put plastic in colonies. And if you think about the interior of a tree, it's really rough and grooved. And that seems to be what stimulates the bees to tack on these resins when the surface is really rough and grooved. And so we wanted to mimic the inside of a tree cavity. So we started taking a wire brush and roughening the interior walls like these photos, and the bees put a little bit of propolis in these roughed wire brush roughened up boxes, but not as much as they would put in a tree cavity. Commercial beekeeping supplier made us some boxes with these ridged interiors, but they were really smooth. And again, the bees put propolis in those gaps, but not very much. And then this guy, Christian Dahm, who no longer is doing this, but he had a propolis hive company north of where we live. And he made these boxes with very rough cut grooves, like you could see in the upper right-hand photo. So there were almost 1/4 inch grooves, just like the propolis trap depth or width, and they were very, very rough cut. So you can see these boxes. There's 2 stacked in the middle photo and 1 on the lower right. And when you put bees in a box like that, they really stuff the cracks with propolis. You can see they still put it in the frame rest, but they really added a lot of propolis in those grooves. And if you leave it in there longer, they're just fill those gaps. They just love to fill that 1/4 inch gap with propolis. So a study that Mike Simone Finstrom led with my graduate student Maggie Shanahan, in collaboration with the ADs, which are commercial beekeepers. They run thousands of colonies. We gave them 60 rough boxes. Actually, we gave them 120 rough boxes, so enough for 60 colonies and 2 deeps. And compared those 60 colonies with 60 others in their operation that did not have rough colonies, just regular Langstroth boxes. And Maggie also ran a part of the study where she compared smooth wood in colonies we just left here in Minnesota with the roughed grooved ones on the right, and then the propolis traps that we tacked in inside the nest that I mentioned previously. So she compared these 3 methods, and Mike, in collaboration with the AD commercial beekeepers, looked at the smooth wood versus the rough wood, not the propolis traps. So 120 colonies, 60 rough groove boxes, 60 standard boxes, and the ADs took their colonies on their standard migratory route from South Dakota over to almonds and then back to Louisiana or Mississippi, it looks like, and then back up for honey production in South Dakota. And they ran them for 2 years, and we followed and took all kinds of measurements as they moved their bees around. So some data from that. The stationary are the colonies that Maggie was looking at, and the migratory colonies are the ones in the AD's beekeeping operation. You can see in the stationary apiaries, the rough boxes, when we were able to score the propolis, the amount of propolis they had, we can score, but we took photos and then scored them. They had significantly more than even with the traps. And in the migratory operation in year 1, you can see in August and then in February, they increased. And then by year 2, they really had a lot more. But it took the migratory operation, it took them 2 years to get as much propolis in the boxes as it took in 1 year in Minnesota. And that was because of the land use surrounding the apiaries. So if you see that first colored bar where it says stationary, that was the land use around our colonies in Minnesota, a lot of herbaceous and woody wetlands, some forest and shrubs and pasture, as opposed to mostly grass and pasture where the ADs were keeping their colonies. Probably not as many propolis producing or resin secreting plants and trees in the area. So it just took them longer, but they got there. When we measured the population size by frames of bees, because a lot of beekeepers wonder if the bees are collecting propolis, will that affect their population size and their honey production? So here's population size in Minnesota, the stationary colonies it made, they all had the same number of frames of bees. And in the migratory populations in August, they were the same. And of course, the populations drop by February. And remember, this is when they get moved into the almonds. And at that time, there was almost 2 frames more of bees in the colonies with the rough boxes that filled with propolis compared to the control boxes, just normal Langstroth boxes. This is important for the commercial beekeepers because those 2 frames are money in their pocket. When they move to almonds, they're judged. They're on the number of frames of average number of frames of bees they're bringing into for pollination. So having more bees at that time is money. So it doesn't look like much, but it was a really surprising finding to me and a good one. Oh, here it is. The data again, just the migratory that year 1 and year 2 combined in February. Honey production was very interesting to me. So on the left is what our colonies produced in Minnesota that year. Very low honey production that year, but no difference between them. And year 1 was the same year in the migratory operation. And you can see, it looks like the colonies with the rough boxes produced significantly less honey than the controls, than the Langstroth boxes. By year 2, there was no difference between them. And we dug into the data a little bit more on that year 1, and we separated out the colonies, whether they had a large, colony, high number of frames of bees, or they were a small colony. And if they were a large colony and the propolis did not affect their honey production, the 2 bars on the left, but if they were a small colony, the propolis did seem to affect their honey production. And in fact, the larger the colony is, the more propolis they had. So these are the same large colonies, and they produced the same amount of honey, and they brought in more propolis. So it's a little bit nuanced, a little bit confusing, but again, I'm going to go back by the year 2, those same colonies, after they had filled the interior of the boxes, all those grooves, the rough grooves with propolis, it did not affect honey production the second year. We also found a really enticing result, which is that there seemed to be a lower amount of European foul brood in the colonies that had rough boxes. This was both by gene expression and signs of disease within the colonies. And Mike Simon Finstrom and others in the USDA lab in Baton Rouge are following up on this study. I hope that's repeatable. For Varroa, unfortunately, we saw fewer mites, not significantly fewer. And if you look at the y-axis, where all of these are under 1 mite per 100 bees. So these are really low mite levels. And there's really, propolis doesn't seem to have any impact on Varroa mites. However, again, Mike Simon Finstrom is repeating some of these studies in Baton Rouge, and maybe he'll come up with some exciting findings. I don't know. All of the data that we collected in the commercial apiaries revealed a very consistent story from all the other studies we had done at the university. The propolis stabilizes the immune system, the microbiome, and the nest environment. And I don't need to go into all of these studies, but this is just some data from all of them. And in conclusion then the use of these rough interior hive boxes to stimulate construction of a propolis envelope is really good for bees. I hope I've gone through this very quickly. I want to thank funding sources, of course, and all the students and people that helped with this work. I thank the cottonwood trees. When I've given this talk before, Native Americans have come up to me and said, of course, we use cottonwood tinctures as medicine. We've been doing this for many, many years. So of course the bees would find the cottonwood and use it for their own medicine. and thank the bees. And I will, I guess, leave it there, or
Okay,
I
so,
stop share. Well, I'll just leave it like this and
okay,
take some questions so I can go back into my slides as needed. Is that all right?
yeah, that was great. Thank you so much. So we do have some questions.
Sure.
Looking at your screen, if I bring these up, can you see them?
Oh, yeah.
All
Nice.
right. Well, why don't, so yeah, thank you, Dr. Spivak. That was awesome. So we'll just maybe attack these from the top and then Kurt kind of takes them off of the question and answer sheet as we go through them. So let's maybe attack this from the top. It says, what impact does bleaching have on propolis when attempting to sterilize hive woodware?
Well, I guess you wouldn't need to bleach your colonies if you had a propolis envelope, because the propolis would be doing the work of the bleach. So, and if you try to put bleach on the propolis, yikes, I don't think that that's a great idea. I don't know what it would do, but it would be unnecessary because the propolis is already doing the work that the bleach is, you're wanting the bleach to do.
In fact, the propolis is probably doing a better job than the bleach is.
Yeah, I would not like to put bleach in a bee colony, but I guess you air it out.
Yeah. Okay, good. How do acids impact propolis?
Pardon me?
So the other question is, how do acids impact propolis?
Huh. How do acids? I guess I need more information on why, where the question's coming from, like which acids.
Yeah. So we'll park that one and then come back to
Yeah,
it if
come
we get.
back at me on that one. And
Okay.
unfortunately, we have May Birnbaum's on this call, so she can help me with that chemistry if I can't answer the question. But let's
Yeah,
come back to it when I understand where the question's coming from. Thank you.
you bet. So this other one is a good question. It says, how long does the propolis retain their benefits? Do bees replace it over time, or
Yeah,
do they have a way to reactivate it when it does, or does
yeah,
it stay active?
yeah, that's an interesting question. So Renata Borba, let's see, second on the left at the bottom, asked that question too. And what she did is collect a little bit of propolis from colonies in October here in Minnesota. And she went to the lab and looked at their antimicrobial activity against some bacteria, and then went back to the same colonies after winter in, I think it was March or April, and the same colonies, and looked at the activity again. And it was less than it was in October from the same colony. So, propolis is releasing volatiles, maybe not so much in really cold weather, but the volatile fraction is part of that's part of the activity is coming from the volatile fraction. Other parts are coming from contact with it itself. So it will lose some activity over time. And by the next season, when the bees start foraging again, they just bring in more propolis and stick it right on top of the old stuff and then keep on going. And the activity, the fresh propolis will be active again. If it's in your way, if you have a lot of propolis in the colony, in spring, early spring, you can scrape it out but encourage the bees to bring more in over the season.
Yeah, that's good.
Uh-huh.
So we have a question here too. In your talk, you referenced sort of the commercial aspects of propolis, not commercial beekeeping, but on the market shelf, right?
Uh,
And so we have a question from Dom Wolfgang who says, does propolis have antimicrobial effects in humans?
yes, quite a few. And in fact, right now in Brazil, there's a 3-day conference on all of the antimicrobial properties of propolis for humans, all the medicinal value for humans and even livestock. 3-day conference. If you go into International Propolis Research Group, I think it might be too late to register, but maybe you can slip into the meeting if you're really interested. But people from all over the world are studying the benefits of propolis for humans, for cancer, tumors, a lot of oral hygiene, oral infections, all kinds of things. It's fascinating.
Yeah, so you said that that it was good treatment for, was it strep B? Or group B strep? I
For
remember.
for strep throat.
No, that you had mentioned that it was effective on group B strep strains. And so where I was going with that is there so if it's effective in humans, then it also has a benefit in veterinarian medicine as well.
Right, right.
Because we keep a kennel here at our house. We raise Labrador retrievers and German wirehair pointers. And it's interesting because some of our pups on occasion get what they call in vet world a strep tail. And so they lose hair and they get a little scabbiness on their tails. And
Oh,
our vet tells us to go out and scrape a little bit of honey out of our beehives and wrap the tails in honey. And it's amazing. Like in 3 days, it's gone.
oh, that's great. Okay, good. I'm glad you brought up honey because I forgot to. So that's really awesome. So the honey itself has a lot of medicinal properties and helps in wound healing and preventing bacterial growth. But May Birnbaum, also a postdoc and people in her lab, found that that honey contains some of the compounds that you would find in these plant resins. They're not coming from the nectar. The compounds are actually coming from propolis that are getting into the stored honey inside the colony. So in addition to just the properties of honey itself, they get like value added benefit from some of the compounds from propolis. I'll call it infusing into the honey. So yes, putting it on the tail of your cat would have probably all kinds of benefits. I am not a physician or a veterinarian, so I'm out of my league here in talking about these kinds of things. If I'm happy to talk about bee health, but I'm a little out of my comfort zone talking about human or veterinary health.
Yeah, well, we know it works from our perspective, so it's a
That's great.
good antiseptic, for our animals and ourselves. We use it on ourselves for scratches and stuff like that. So we have a clarification on the acids
Great.
that we were being referred to. So the acids like oxalic acid or formic acid.
Oh, I see. I understand. Okay. So the question is, how does propolis interact with the mite treatments?
Yes.
I don't know actually the chemistry of all of that, but I think they would be okay to use. Oxalic is a contact with the bees themselves. So if, I've only used the dribble, I've never vaporized with oxalic, but it just gets on the bees. And the formic acid, the beekeepers were treating the commercial guys were treating their colonies with formic and other compounds, and oxalic. And, I really didn't think to ask the question how the propolis might affect the treatment, but as far as I know, it did not. And if anybody listening has more to offer about that, please weigh in. Yeah, thank you.
Okay. So the next question, are there direct measures on antiviral activities from the propolis, since you stated that it had some benefit, it looked like with
Yes.
HIV?
There have been some studies from a woman in Europe, Drescher was her last name, that did find colonies with added propolis in them had lower amounts of deformed wing virus. We didn't see it in any of our studies that the viral load was lower. You would think that it would. And I think that Mike Simon Finstrom in Baton Rouge is doing some more studies on that. But we didn't see it in our data, in our studies.
Great. So here's one. Will the propolis envelope increase the quality? In other words, the nutritional value of the honey? And we touched on that just a minute ago, but
Oh,
anymore.
I would say yes, but that's just based on my bias about propolis and honey in general. May, do you want to add in on this one?
Hi, I'm working on the last one. Just to talk about acids. Actually, resins are loaded with various and sundry forms of acid, phenolic acids, for example. But what was the question about the oxalic acid? Is that right?
Yeah, I think the question was, Maze, does propolis affect the effectiveness of, say, oxalic acid or formic acid? Does it affect it one way or the other?
I have no data that would be relevant for that. And I think Marla's, I don't know if it's speculation or not, is probably as close as anybody is going to get, because just putting in a pitch in here, Marla has busted the world of propolis wide open. For years, it was just ignored, scraped and tossed away. And you really laser-like focused attention on what is clearly very important for the life of the bee. So bees are the most amazing chemists. And one thing that just I can't get my head around is how they know which trees to visit or which plants to visit for resins, because they unerringly choose the ones that appear to be the most beneficial in terms of health, and that defies human skills. I can't really predict where they're going to go.
That's, they're not always choosing the best resins, interestingly. Well, I don't know if I can really say that. So when, I think they're using odor to locate the, if a cottonwood tree while it's leafing out, which it does all season long, just smells amazing. So I think they're using odor to locate the trees. But when we collected resins from all kinds of trees in our area, and this was the royal we, I'm talking about Mike Wilson, graduate student, and looked at the antimicrobial value of, or properties of different resins, he found that the most antimicrobial resin was white spruce. And honeybees are not collecting propolis from white spruce, at least in our area. The populace was somewhere in the middle compared to some of the spruce and pine resins, but they're not collecting. And I think it's just abundance. And often the conifers that produce resin do it only at certain times and if they're wounded. So it's a response to being wounded, and a cottonwood tree is producing it constitutively just all the time. Every leaf bud has resin on it. So I think, honeybees love to shop at Costco, and I think they're doing the same thing for cottonwood trees and resin.
Although they'll also, there's at least one study showing they pick up road tar. So they don't,
Yes.
right?
Yes, they're not very discerning completely. Yeah, that's true. Another question, though, can we go back to it, is concerning the honey. And can you remind me of the question, Scott, about honey and
let me.
value added?
Does it add nutritional value to the honey?
Does a propolis? Can you answer that one for me, May? This is
well,
one having.
Not nutrition per se, but there's the concept of well, sort of around 30 years ago, it was introduced into the nutrition literature called functional foods. So these are foods that don't contribute sort of classical nutrients, but are health promoting. And in fact, that's one of the most important things that a lot of bee products do. Even if they don't add nutrition, their phytochemistry does have health promoting benefits, in that it, in addition to being antimicrobial, some of the constituents of propolis and honey turn on or upregulate detoxification genes. So this is a way that humans and bees deal with various and sundry toxins in their environment. So it's another thing that, don't take propolis away from bees, because it's actually helping bees cope with not just their biotic enemies, but their inorganic enemies that, like humans, contaminate the environment with, like pesticides.
Yeah. Thanks, May. I really didn't pay May to come to this
Oh,
meeting.
and I didn't come to butt in either, so.
All right, well,
Yeah. So Marla, there was a clarification on the oxalic acid, formic acid question that we had.
okay,
I'm
sure.
trying to find it. Just a minute here. I'm gonna scroll through it. Okay, so let me get to the top. Sorry, but I can't find it. I think it was, does the oxalic acid or formic acid affect the propolis itself?
Oh, yeah, I really don't know. Sorry.
Okay. All right, well, let's do this one then. So how would you encourage bees to deposit propolis in plastic thermal hives like styrofoam?
Oh, boy. I think you're gonna have to tack in a mesh, a hard mesh inside the walls.
Yeah, I think it shows also, like you were talking about, the inside of a smooth Langstroth hive box. They're not, except in cracks and corners, they're not
Right,
propolizing the inside of that, which is a smooth surface as well.
right.
How should beekeepers clean out the bee droppings inside rough boxes?
Clean out the bee droppings.
Yeah, it looks like if they're pooping inside their hive.
Oh, I think you can just go ahead and scrape as you need to, and clean as you need to. And as long as there's that rough groove still in there, the bees will, after you put it back on the bees, they'll just continue to add more propolis in there.
Yeah,
So it's not that you do what you need to do. You can treat it as a regular hive and scrape as you need to.
yeah, that may be a tag on to the bleach question we had is, if you're cleaning poop out of the inside of your hive with a bleach, what does that do to the propolis? But not that we need an answer to that. I'm just saying maybe that.
Well, you know that propolis studies in Brazil that I didn't show here at all, but the propolis is having some activity against nosema also. And I think in that
Right.
study, we're feeding the propolis in sugar syrup. If you can get it into solution, it's very hard, but they were feeding it to the bees and it was helping with nosema. So maybe having propolis in the hive, you wouldn't have dysentery or nosema as much. I don't know.
Yeah. Yeah, if they're pooping in the hive, that's a problem.
Yeah,
Right inside
that is a
there
problem.
in the house. So
Yeah.
did the colonies that the boxes where the propolis painted inside add to that propolis?
Yes, you can paint the propolis. You can make this extract or tincture and paint it. But it's just a lot better to have the bees do the whole work for you, because to make the tincture, you've got to collect the propolis, then you've got to dissolve it in ethanol, and then you've got to figure out what percent solution you're using or not, but then paint it back in the boxes. And yes, the bees will continue to add some more resin or propolis on top of that, but it's just better, I think, to have them collect fresh and local propolis directly from the trees and put it right into the box rather than having to do it yourself.
Yeah. All right. I think we may have answered this one already, but I'll say it again. Does the quality of the propolis degenerate with age? And I think you said it does. And then that the bees kind of refresh it with new propolis on top.
Right.
Yeah. So let's see. If hive bodies were made of rough cut or roughened lumber internally, do you think the bees would propolize the rough surface without not surface, but rough surface without encroaching on bee space?
Yes, they will. They'll just fill it, they'll just put a layer right on the rough surface. If it gets in the way of the bee space, you can scrape it off.
Yeah.
So it won't be a quarter to a half inch thick. We're talking about an 1/8 inch layer. And if it's grooved, then they're going to mostly stick it in the grooves where it would not affect the bee space.
Yeah. So we have another question here, and some of your audience tonight Marla, or international beekeepers, so they're, not from the United States necessarily, so they're reaching out with some questions that are,
Yeah, that's great.
that are, unique to their circumstances.
Sure.
And so this is a question probably for a good, starting beekeepers, and that is, does Propolis and Nosema, how does it affect the beehive?
Well, like I just said, that some studies in Brazil where they were, and also maybe in Uruguay, where they were feeding bees propolis dissolved in sugar solution, that it did seem to have an effect on nosema in the bees. It was help treating it. I don't know if it eliminated it, but it helped. It was seemed to be okay as a treatment for nosema. Now, I'm not sure I would like to feed propolis to the bees. That's not how they're consuming, that's not how they're using propolis naturally. It's contact and volatile, that's their activity. Consuming it would be like taking a very, very strong antibiotic, I think.
Yeah. So probably good to rely on the more typical nosema treatments than using propolis for that, right?
Well, I would have to review the literature a little bit more to really to weigh in on that one, but yeah.
Okay.
Yeah.
All right. So the next question, is there a market for propolis collected?
Yes, there is. In the US, though, it's a little bit tricky. We found a place in Oregon that would purchase it if it was very, very clean propolis. I'm not sure about Canada and other places, but I wish there was more of a market. Part of the problem is that people from the US don't use a lot of propolis products for their own health. If you go to South America and you walk into any store, Walmart even in Brazil, you're going to find a whole half an aisle on propolis products for humans. So there, the market is huge. So it just depends on what country you're in.
Yeah. So another question is, what race of bees collect more propolis? And I think you mentioned Africanized bees, or?
Well, Africanized bees in general collect more than our European derived bees, but within our European bees, it's, the Caucasians were notorious, had a reputation for being big propolis collectors. But from the bees that I've seen, you can find propolis collecting bees within any stock of bees that you can find, Carniolan or Italian derived, or any of them. You'll find some colonies that collect a lot and some that don't collect as much.
Yeah, that's okay. Does raw and unfiltered honey have small deposits of propolis that would have microbial benefits?
Yes, that was the research that May students did. Yeah, May, and they did in her laboratory. Not all of the propolis compounds were found. My understanding is that some of the compounds from propolis you could find in honey.
Yes, we had a follow-up question on feeding bees propolis, and that was, can you feed bees with propolis? And I think you probably answered that one already. Would the propolis at the entrance door, would the propolis at the entrance work like a doormat, clean the bees off a bit?
Yes, yes, I think so. I always wanted to set up an experiment to have one of the treatments would be just to have like this propolis doormat and to see if that would be enough as bees are going in. Would it have the same effect on the immune system and et cetera? But we never ended up doing it. But yes, they do tend to put a lot thicker layer right around the entrance. I don't know if it's just to help reduce it. They could use wax to reduce the size of the gap, but they're using a lot of resin in it. Makes me wonder if it has particular, an antimicrobial doormat. It's a nice addition to everybody's house, right?
Yeah,
Wipe
that's
your feet on the sticky mat.
the way they take a shower maybe before they come in the house.
Yeah, yeah.
Will any outside insulation installed to the hive hurt or affect any propolis that may be installed? So, or maybe propolis inside the hive.
I think the question was any out external insulation for the
Right.
winter affect the propolis inside? No, no.
Great. How do you clean propolis?
So I'm assuming you're saying if you scrape it from a colony,
Yeah, I'm thinking.
and it has bee parts and wood parts and beeswax in it. Well, it's very difficult. We
Yeah.
don't. So when we are collecting propolis, if we're trying to sell it, it's straight from a propolis trap that
Yeah.
we've put on colonies. So it's very clean. It's just almost straight resin.
Yeah. So this is an interesting question. Is propolis influenced by soil health? In other words, the quality of soil microbiology. Is there a connection between that?
Well, my guess, I don't know. It's not so much soil quality, but by stress in the plant. So, and I'll just give this story. I heard in Brazil, this baccharis shrub that produces the green propolis. I showed a photo early on in the talk about that.
Yeah.
This is a propolis with lots of antimicrobial properties that they've done a lot of research on. So they planted a whole plantation of this shrub, and they watered and took very good care of it, good soil, and it produced very little resin. But out in the wild, where it was a little bit stressed and needing protection against other insects and herbivores that would eat it, or other conditions, that's when the plants are producing these resins as their own defense. So I guess I don't know if it would, I think the plants would produce more resin when they really need it as a plant defense, no matter what the soil is. But this is a guess, I'm just speculating.
Yeah, yeah. Okay, so is the propolis in the honey making honey antimicrobial? And I think
Yes.
you said that it is.
Yes,
that.
May, do you want to jump in? You're invited.
Sure. Well, honey's pretty antimicrobial on its own. I mean, there's the whole phenomenon of, by which honey produces hydrogen peroxide, which is a sterilizing agent. So many of the nectar flavonoids that end up in honey when the nectar is concentrated down themselves are antimicrobial. So yeah, honey is remarkable stuff, and it is definitely antimicrobial. Of course, there are lots of microbes, but there's so there's the peroxide derived antimicrobial activity, and also there is activity based on nectar constituents. So honey is amazing stuff.
Yeah, we had Scott Dednam on last month talking about thermodynamics, and he made in the hive, and he made a great comment. He said, honey has more benefit than just sweetening your tea.
Oh, absolutely. In fact, if you read the tabloids, you'd think it cures everything, but that's a little bit off point. So.
You bet. All right, here's our last question, which is, when I melt my uncappings in a wax melter, is that propolis residue that is left?
Yeah, the slum gum contains some propolis, but also other junk, who knows, but it's not pure propolis, so don't, I wouldn't use it for that. It's really good fire starter, slum gum. You mix it in with a little dryer lint and light a match to it, and you can start a fire right now with it. It's great stuff for that.
Yeah, you can use it in your smokers.
Yeah.
Well, Marla, that's all the questions we've gotten, and tell you, it's been awesome. So thank you so much. So we had one that came through the chat, and that is, how do you separate propolis from honey? I think it was, but
I don't think you can.
I don't know that you can, yeah,
Yeah.
okay. Well, I think we'd be remiss, Marla, if we didn't say that your work is with Propolis is kind of redefined beekeeping in a lot of ways. So thank you so much for what you've done over the years. You've been
Thank you for having me, and
a great blessing to this industry, and we love you for it. So
and
thank
look
you.
at all the little emojis floating up.
Yeah,
Thanks, you
people
guys.
love you.
That's sweet. That's wonderful. Thanks, May, for it was wonderful. It's fun to. I don't get to see May very much. She's at University of Illinois, for those that don't know, and has also done a lot of great research. There she is. Thanks. Thanks for
There's
waiting.
May,
That
yeah.
was wonderful for me.
All right, well, for those of you that are visiting here tonight, and if you go on the WASA's website in a few days, then you'll be able to see this again. Sure, you want to review it a few times, and there's also, do a little pitch here, a registration form if you want to join our organization. It's $30 a year, and this alone tonight was worth every penny of $30. So,
Thanks.
all
Thank you.
right. Well, thank you, everyone. We'll see you next month. We're going to be talking about mite-eating bees at our next mini conference. Have a good night, everyone. Thank you.